The variable chains of lymphocyte antigen receptors are associated with invariant accessory chains that carry out the signaling function of the receptor. The antigen-binding portion of the B-cell receptor complex is a cell surface immunoglobulin that has the same antigen specificity as the secreted antibodies that the B cell will eventually produce. Indeed, it is identical to a secreted monomeric immunoglobulin, except that it is attached to the membrane through the carboxy termini of the paired heavy chains. The messenger RNA for the cell surface heavy chain is spliced in such a way that the carboxy terminus of the protein is made up of a transmembrane domain and a very short cytoplasmic tail. The heavy and light chains do not by themselves make up a complete cell surface receptor, however. When cells were transfected with heavy and light chain cDNA derived from a cell expressing surface immunoglobulin, the immunoglobulin that was synthesized remained inside the transfected cell rather than appearing on the surface. This implied that other molecules were required for the immunoglobulin receptor to be expressed on the cell surface. Two proteins associated with heavy chains on the B cell surface were subsequently identified and called immunoglobulin alpha and immunoglobulin beta. Transfection of immunoglobulin alpha and immunoglobulin beta cDNA, along with that for the immunoglobulin chains, results in the appearance of a B cell receptor on the cell surface. One immunoglobulin alpha chain and one immunoglobulin beta chain associates with each surface immunoglobulin molecule. Thus, the complete B cell receptor is thought to be a complex of 6 chains, 2 identical light chains, 2 identical heavy chains, 1 immunoglobulin alpha, and 1 immunoglobulin beta. The immunoglobulin alpha and immunoglobulin beta genes are closely linked in the genome and encode proteins composed of a single amino-terminal immunoglobulin-like domain connected via a transmembrane domain to a cytoplasmic tail. Immunoglobulin alpha and immunoglobulin beta provide the only substantial cytoplasmic domains present in the receptor complex and are crucial for signaling. The transmembrane form of the immunoglobulin heavy chain has a very short cytoplasmic tail, and it was hard to understand how this could signal into the cell that the surface immunoglobulin was ligated. The discovery of immunoglobulin alpha and immunoglobulin beta solved this intellectual problem. And that's the variable chains of lymphocyte antigen receptors are associated with invariant accessory chains that carry out the signaling function of the receptor. Have a great day.